Good afternoon, everyone. I'm Forrest Ray, reporter with Genomeweb, and I'll be your moderator today. The title of today's webinar is Accelerating Outcomes in Pooled CRISPR Screening Data Analysis and Drug Discovery, and is sponsored by Seqera Labs. Our speakers are Miika Ahdesmaki, Senior Director of Early Data Science and Oncology R&D at AstraZeneca, and Evan Floden, CEO of Seqera Labs. You may type in a question at any time during the webinar. You can do this through the Q&A panel, which appears on the right side of the webinar presentation. If you look to the bottom of your window, there are a series of widgets to enhance your webinar experience. With that, I'll turn it over to Miika Ahdesmaki.
All right, thank you so much. So thank you for tuning in today. So I'm going to talk about functional genomics and CRISPR screening, data analysis, data science, and how that helps oncology drug discovery and uncovering drug resistance. So some of the challenges we see in oncology research is drug resistance and also predicting effective and safe combinations. So you can see on this images there's a patient with melanoma that is BRAF mutant, and initially they respond really well to a BRAF inhibitor, but then eventually the tumor becomes resistant. So despite treatment, the treatment no longer helps and the patient relapses. So also productivity and long development times in drug discovery are some topics that are really key to address. And some of the solutions that I'm showing here today is to understand drug resistance and predict effective and safe combinations. One approach preclinically that can be done in the discovery space is something called pooled genome-wide drug CRISPR screens that I'm going to talk about today. And in terms of productivity and shortening development times, data analysis is key. So data science and great data strategy are key to address productivity. And this includes data and algorithms in the cloud. And today with Seqera talking about, for example, how to orchestrate pipelines using Nextflow and Nextflow Tower. So in terms of functional genomics, a CRISPR, in this context, there are these CRISPR gene editing libraries that you can buy off the shelf. And what you could do with these libraries in preclinical models is you can knock out genes, and that's the focus that I'm going to talk about today. But you can also do transcriptional repression, that's called CRISPR-I, and also turn and switch genes on, also known as CRISPR activation. But my focus today will be on CRISPR knockout. So that's basically knocking out genes and seeing the effect on the cell. Now, there are different applications of CRISPR screening in oncology. So you have, for example, de novo target discovery, but also understanding drug resistance and receptor expression. So what I'm showing here in the subfigure A is you take a pool of cells and then you do your CRISPR screening. So let's say you're targeting 20,000 genes and maybe your library has 100,000 guide RNAs. So what we do in this setting is you are looking at when you knock out genes, the knockout of which genes causes cell death. And this kind of data is available online, so you can go to the dependency map portal and you can actually download this data if you're interested in target discovery. But another application is when you perform this CRISPR screening with a control arm. So that's similar to subfigure A, but additionally you compare to a compound. So this compound could be a drug in development that you're interested in knowing preclinically what might make your cells resistant or equally more sensitive to your drug. So using this approach, you use CRISPR to knock out all the genes, and some of the genes will come as what we call as hits. So you might have 5 genes that when you knock them out, the cells no longer respond to the drugs, or you might have another set of genes that actually the cells become even more responsive to drugs. So you're sensitizing the knockout of these genes, sensitizes the cells further to the drug. So those could be combination hypothesis genes. There are also other approaches that I'm not going to talk about today, but there are single cell approaches now the CRISPR. So you might have a CRISPR library of, let's say, 10, 20, 30 genes, and then you also observe the single cell transcriptome of the knockouts and see what happens. And also FACS CRISPR. So what you can do with FACS CRISPR is you build this similar to subfigure A, you knock out all the genes, and then you'd use FACS to sort the cells into sort of high expressors and low expressors of something, and you can understand what is it that actually the knockout of which genes modulates that cell surface receptor, for example, HER2. Now, at AstraZeneca, we have something called AstraZeneca Cancer Research UK Functional Genomics Centre in Cambridge, UK. It's a joint venture for running CRISPR experiments at scale. So we have access to different kinds of CRISPR vectors that I mentioned. I will mention more on the next slide. Where we're based is actually on this Cambridge Biomedical Campus. It's a very vibrant campus in Cambridge on the Addenbrooke's Hospital site, and it's right next to the AstraZeneca Discovery Centre. We have different kinds of CRISPR screening technologies there for novel target identification, and between the two organizations, we do joint development of vectors, models, and also computational pipelines. So then just looking a little bit at the different kinds of options for CRISPR screening. So you have different libraries. So you can have, let's say on the left, genome-wide, one gene knockout libraries, but we're also building multiplex gRNA libraries. So you might be able to use two guides in a construct to hit two genes at the same time, or hit same gene in two different spots. And also, if you've heard recently, there's something called a lot of excitement about this prime editing that enables more precise editing of the genome. And in terms of model systems, there's the usual 2D cell lines, but also nowadays 3D organoids, immune cells, and in vivo models. And then if we again go back to what can be done with CRISPR screening is in the drug CRISPR screening, we're looking at different ways to make cells resistant to a drug or sensitize the drug, so get these combination hypotheses for new targets. And I also mentioned this FACS screening, where you can understand the modulation of cell surface receptors and single cell RNA-seq combined with CRISPR. And then very importantly on the right, we've got different computational approaches that are required here to process all the data and get those insights. So what we have is a cloud-based pipeline and different algorithms running in the pipeline, so we get a comprehensive view of what we believe to be the statistically important screening hits, and then different kinds of enhanced interpretation to bring context to those hits, so the biological context into the CRISPR screening hits that come there. Okay, so I mentioned this fair data and data strategy. So fair means findable, accessible, interoperable, and reusable. Many of you will have heard that term before. So because we run this large center for these studies, we haven't run one study, we've run tens of studies. And the power of running tens of studies across different kinds of molecules and cell lines is you can then collect this into a database and start looking at whether you see patterns what are the patterns that appear across different kinds of studies. Now, we didn't want to reinvent the wheel, so we looked at what's out there in terms of database design, and we found this BioGrid open repository of CRISPR screens, or ORCs, that we borrowed the schema from. And what this does is it gives us a holistic view of CRISPR hits from these studies that we've done, drug CRISPR studies we've run, to identify these pan-study patterns of drug response. So I'm just giving here an example of a study that was run where this was actually a CRISPR activation. So what the results showed us that is there's actually a gene called this BRAF itself. So if you have an EGFR, there was a study of EGFR inhibitors, and if you activation of BRAF in this setting actually caused resistance. But also we found that when we looked at across different kinds of public data sets of CRISPR data set, that BRAF was coming as a hit in these other studies. So you can start putting the picture together of what does the result of a single study, what does that look like in the wider context of other studies. And then it's just there's the obvious question come from when you start integrating this data is, do we, across very different kinds of molecules across different kinds of indications, whether there are some genes that always cause resistance, regardless of your molecule and regardless of your cancer type. And the answer is no. When we look at this across these studies, resistance genes are that they only pop up across, let's say, what it says there's only 110 genes are hits in at least 5 studies, but nothing is really a hit across more than in the 20 studies. So what this is saying, there's no silver bullet gene that would always cause resistance or sensitize. Okay, so what I'm going to move on to next before handing it over to Evan is look at a little bit under the hood of what this analytics looks like in the cloud, and is facilitated by Nextflow and Nextflow Tower. So what we do within the Functional Genomics Center of AstraZeneca and Cancer Research UK is that the lab team works as one. So we've got one lab team from both organizations together, and running these screens with the different kinds of molecules. But when it comes time to sequencing and data analysis and insights, then we split. At that point is where it becomes proprietary. So AstraZeneca data goes to sequencing and comes back to AstraZeneca firewall, and is analyzed within our firewall using this, but using still a shared pipeline with CR UK. So as part of running these studies, what we need obviously the sequencing data, but we also need a lot of information from the lab in terms of what were the cell lines and what were the treatments, what were the time points, and all of this is quite complicated in the end. So you get a lot of data. So to drive the pipeline and make as few errors as possible, we'll work with the lab to normalize these, validate these fields, and also make sure that all of this is automatically translated from the way that they capture this data in the lab into something that can drive the analytics pipeline. And in this case, the analytics pipeline is driven by a JSON file. So we're converting this tabular information from the lab in an automatic manner in the cloud to something that drives the pipeline, so that we don't have any manual intervention that could introduce errors. And here's just a snapshot of some of the higher level complexity of the pooled CRISPR screening pipeline that we use. So on the left, sort of left top hand side, we see that we have data ingestion from a server coming into Amazon Cloud. So the data is coming from the sequencing provider, and it comes into the cloud. And in the cloud, we then combine that with what we call the experimental metadata. And all of this runs in an automated manner. So if it's a standard project, there's actually no manual intervention. Where there's a signal or push notification coming from the sequencing provider, then the data download kicks off, and then the metadata that has been captured in the lab, that gets translated into the JSON format, and it drives the Nextflow pipeline that's in the middle. And from the Nextflow pipeline, when that's run, then the results go in this database that I mentioned, where we can look at the data across different studies. But also we have Nextflow Tower to help us with pipeline monitoring primarily, because the launch is automated. And then we also we built a QC dashboard, because if we don't understand the quality of the data, then obviously we'll be flying in the dark. But the powerful Power BI, we use that to understand what is the quality, not just within the experiment, also across experiments, if there's any kind of trends or shifts in the data. So in a normal pipeline run, if we want to look under the hood, I mentioned this JSON file, so we really have to just specify the JSON file and have the data in the right S3 bucket, and then the pipeline kicks off automatically. But this can also be done manually through Nextflow Tower. If there's something needs changing, manual changes in the configs, then we can launch this in Nextflow Tower. That's a very nice feature. And then what we like about Nextflow Tower is that it tells you where you are in the pipeline, how many processes are running and how many are finished, and what kind of time it's taken to run the pipeline. But also it tells us what are the bottlenecks. So in this case, we're looking at what is the CPU and the memory bottlenecks, and where should we be optimizing. So if something takes a lot of CPU, can we optimize that part to and not focus on things that are actually insignificant in the grand scheme of things. And when eventually the inevitable happens that there's a problem with the pipeline run. Maybe there was some problem with the metadata, or maybe some of the data was incomplete. The good thing is that we get this error from Tower, and it helps us then go and debug the pipeline and debug the error, fix whatever caused the error, and then go and relaunch the pipeline and get the results that we need. So this is in short what we do in the Functional Genomics Center. So we use this pooled CRISPR screening approach in genome-wide to understand drug resistance and also come up with new hypotheses for combinations. And I talked about this improvements in productivity. So without this automation, all of this automation, and before we built this automation, it used to take days to set up these runs manually for the analysts. But obviously, because at that point, you have to make a decision, is this a project or does it need a process? So when you run something repeatedly again and again, it's worth building a process. And the nice thing with Nextflow and Nextflow Tower has been that we've been able to build a very smooth process where we get all the time back for the analysts to spend on the insights, the biological insights from the data, rather than the running of kind of the pipeline. And with that said, I would like to thank all of my oncology R&D colleagues, as well as our R&D IT colleagues who work with us to have over the last a few years to build all of this automation and help us get time back for science. But also thank the Cancer Research UK, Cancer Research Horizons colleagues who we work with together on these pipelines. And with that said, I will hand it over to Evan.
Great, thanks so much. There's lots of really interesting information and material in there. I mean, we've known each other for a few years and obviously been kind of following the project, but it's kind of great to see some more detail on that and you know, how some of these kind of processes are helping out with that. So nice to meet you all, folks. Thanks everyone for showing up. My name is Evan Floden. I'm the CEO, co-founder of Seqera Labs, based out of Barcelona. If you don't know Seqera, we've been around for about 5 years, but the kind of origins date back a few bit longer than that. We created the Nextflow project, so Paulo de Tomaso and myself, about 10 years ago now in 2013. So we've been kind of building the Nextflow project out, and really in the last 5 years been working on Seqera. The idea of the Seqera is really to make data analysis accessible at any scale and building on some of the things that we learned from Nextflow and then kind of taking that to the next step. I think just you know, touching on some of the points made previously, we talked about like, are we ready for having a process for something, or are we just trying things out? I think one thing that we see common across all the customers that we have is there's a really big spectrum of what people are doing. Sometimes you're in situations where you're doing research, where you're iterating, you need to iterate very fast. Sometimes you're in situations when you really need to like lock things down and run things at scale and run things at production. And by really kind of, I guess, using modern software engineering practices, you're able to kind of lean into both of those things. And when you're ready to go from that iterative environment into that production environment, it helps a lot as well. So as I said, at Seqera, we're building the modern biotech stack. And I think stack is a really kind of important point here. We are not building a SaaS platform that you may typically see around. The idea with Seqera, we're really building foundational tools which you can build off, which you can build on top of. And I really liked the last slide there, the second to last slide. We kind of showed all of the different pieces of infrastructure which are involved and which are kind of relevant to how we build out some of these systems. And that integration layer, that ability to have that flexibility to use the tools that folks use is really important. Okay, so enough kind of intro. Start on like a little bit on the origin. So if folks are not familiar with Nextflow, so excuse folks who have been around for a while on this, but we started off primarily focused on the workflow execution. Now, this diagram itself is really kind of showing, in this case, ancient DNA analysis, but it really could be anything. It could be CRISPR analysis, as we were talking about before, but it could be just as be an RNA-seq or variant calling pipeline. The point being that we've got lots of different steps. Each one of those steps is a different piece of software. Maybe it's something we've taken from Bioconda. It's a container we've pulled from somewhere. It's a GitHub repository. It's the postdoc script that you got inherited from a couple of months ago. You have to pull all of those pieces together with very different requirements, with very different resources, into a pipeline and really turn it into a piece of software itself. And this is really what we call a pipeline, the idea that you can take those individual components, you can write them, you can use the Nextflow syntax to define them, and really from there take that and basically kind of run it at scale. So the idea of Nextflow was very simple, combined a couple of things. The first was enabling scientists to write those pipelines. The way we do this is via a DSL, so it's like the Nextflow syntax. It's kind of a very simple way of taking existing code and wrapping it and writing it from there. The other kind of real core piece was the use of containers. And very early on, we saw the use of Docker as it was applied to web services, and kind of thinking, how can we apply that to data pipelines themselves? This is now very commonplace, and you'll kind of see this in our services, but at the time, it was a pretty novel concept, and we've kind of really leaned into that concept of containers. It's obviously essential to reproducibility, but also portability, which I'll get onto in a moment. The other one was really just the adoption of cloud, as people were starting to think about how can you run these pipelines from your laptop to your cluster to really just make use of those resources that were available. How can we really make that available for folks? And I guess the real key to Nextflow has been this interoperability layer. The fact that anyone can write these pipelines themselves and then share them with the world. And it's really that sharing, that open science collaboration, which comes about from that, which enables all of the great content to be built. There's a fantastic slide that we had before where kind of showing that you could take the same pipeline, you could take that same infrastructure, and independent of where you were going to run it in the AstraZeneca VPC securely, or if you wanted to run it in complete other environments, that was completely shareable. And the reason that it was is that because the code, the actual analysis is not really tied to any infrastructure. And that really makes a big difference if we think about the fair values and also the scalability as well. So just touch on Nextflow itself. Nextflow is a language, as I said, it's a way to write these pipelines. But you're typically taking existing code. You're taking your R script, your Python, you're taking your Bash, and you're really just wrapping a thin layer around it to be able to connect those tasks together. When you connect those tasks together, you'll define for each of them a container. Essentially, you'll be able to use the dependencies. Each one of those will become a task, and those tasks will then be submitted into the environment of your choosing. Nextflow handles all of the orchestration. We've also been very big into use of Git. And Git in itself provides a lot of the kind of code tracking, the reproducibility, the versioning that comes about, but it happens automatically with Nextflow. So in Nextflow, you get this automatic use of containers, this automatic use of Git, and that's something we've as we build out the rest of our products, have been continuing to use as well. It's really the separation which is key, though. You can take any Nextflow pipeline and run it in any environment, and then in itself has allowed people to use development, to use modern software engineering practices where you can write local, where you can push to your Git repository, and then you could fire that up in the cloud. You could work with your collaborators independent of the environment that they're in. And this is given the kind of the community aspect that Nextflow has. Moving forward a little bit on the syntax, so just touching on this, it's designed for scientists. So you have existing code. In this case, I've got a Salmon command line tool. Independent of the tools that you're using, this is a command line, but it could be Python in here, it could be an R script, whatever you'd like. You define inputs and outputs, link those together, these tasks together, and then run. What's really great though about Nextflow is that now, particularly with NF Core, we've got over 1,000 modules. So 1,000 of these tasks have already been written for you, and you can really lift them, drop them into the pipelines that you need without necessarily even needing to worry too much about how to write those pieces as well. Again, we're not looking to rewrite everything from scratch. We are reusing those existing tools and kind of leveraging the other work that folks have done, and really just having so many sets of eyes on these pipelines and on this code has enabled you to really focus on the science and not having to go so much into really worrying about the engineering of this. So wrapping up all of that, as we think about Seqera itself, we're really trying to make scientists be able to focus on their science, to be able to run things at scale, run things flexibly as well. So that integration layer, that ability to use the tools that you're already using. This was where we were thinking about integrations with Git very early on, or now as we think about the integrations that we have with containers, with container registries, there's no real need to change the infrastructure that your organization uses or that you should set up. Really, the kind of best practices that folks are using and the rest of the IT industry can really adopt that now and kind of use that towards your science as well. If we think about some of the kind of, as we've expanded out, we've focused a lot on the pipeline execution. This is kind of this concept of data pipelines or workflows. We've done this for a fair amount of time, and it's, guess what Seqera is most well known for, given the kind of history with Nextflow. This is something we continue to expand out, particularly if we think about performance and scale. The scale in particular, when we start to see folks running really large pipelines with significant amounts of data, there starts to be some real significant challenges when they run that. So when you start to, for example, launch into the cloud, you can have difficulties with regards to the storage, which is associated with that compute, with the ability just to like scale that amount or those amount of resources, and also to do that in a cost-effective way. There's lots of incredible benefits of going to the cloud, spinning things up on demand, just like there's lots of benefits of being able to really have your compute scale and be able to use schedulers in an efficient way. But ultimately, we all here, if we think about those costs, they are not insignificant, particularly at scale. And getting those down has been a real key thing that we've been doing. Examples of this and how this worked is really around the optimization of resources, some of the storage technology which we've been building out, as well as really just taking and building the pipelines in a more efficient way and making them available for folks. We like to think about this as the data analysis lifecycle. And depending on whether you're at what I mentioned at the beginning, if you're really into the research side of things, or whether you're much more on the production side of things, will depend on how much time you spend in each of these areas. Whether you are developing code, whether you're developing workflows, you're building containers, doing a lot of iteration, then you'll be spending a lot of time here. For this, we look to things that happen in the community. There's fantastic VS Code plugins. There is ability to build your containers using Wave, which is a technology we've been released recently, where we're looking at to be able to build those tooling with the NF Core tools that have, for example, linting. You can really integrate all of that into your workflows and really be able to produce those pipelines and kind of say leverage a lot of the community side. A product that we have coming up very soon, it's actually available on the cloud product, is what we call Data Explorer. This is a way to connect into your existing resources. What this does is it essentially connects your cloud resources independent of where they are, whatever region, whatever cloud that you've got, and provides you with an interaction based on browsing, based on being able to really connect in, to upload, to download. But importantly, it's not like typical black box situations where you're dropping your data into the cloud. It's visibility on those existing resources that you have, whether this is connected up to instrumentation, whether you want to connect this up to the results of your pipelines. You have that ability to browse, to connect, and really to have some traceability of what's going on. Finally, what we're getting to really excited about is moving away just from having the batch processing, but more into the interactive processing. For us, thinking about this is most likely in situations of Jupyter notebooks, our studio environments, our Shiny applications, IGV genome browsers. The key, though, is that you're having that interactive environment, but that interactive environment is launching where you have your compute and where you have your data. And by connecting those two things together and being able to launch an interactive environment there, you're really avoiding a lot of the hassle which is required with moving that data, with the authentication, with security associated with that. Finally, we're thinking about on building out the reporting. So one tool you may be familiar with that Seqera develops is called MultiQC. It's the most widely used reporting tool. It works with hundreds of bioinformatics tools and collects those logs, collects that information, and really kind of wrapping that information up to look at that across different runs and to bring all of the information that we have associated with runs into that as well. So that's a quick summary of what we've been working on and how we're kind of expanding the portfolio of things that we're doing. I'd love just to touch on a couple of examples of where this has been useful and kind of show how folks are using the technology sort of beyond the examples that we see here as well. So touching on this, in the pandemic, Nextflow was used really widely, and touching on some of those things around use of GitHub, use of Git control, ability to launch in different environments. Scientists were really able to come together. They were able to define essentially new pipelines, new pieces of software specific for SARS-CoV-2. They were able to share that code literally like locked in their bedrooms, like many of us, but really share that code and really get it to the points of sequencing. The UK in particular was very strong in this. They were able to send that out. They were able to sequence it at some places around 20% of all folks who were being tested. They were sequencing and really use that information to drive the kind of knowledge around variants and what was taking place. This has now been replicated in many places, not necessarily for SARS-CoV-2, but more generally in public health. And we see this in foodborne diseases and also really kind of generative weather maps of kind of public health. This is kind of just the beginning of this, we believe, in the use of sequencing in itself for that information and how it can advise us is something fantastic to see. We also see examples in synthetic biology, so development of new materials, really creating really new genes, new organisms to really create those materials as well, and how that can reduce waste, how that can be used in synthetic biology generally. Again, another kind of high sequencing approach where you're kind of using that information. This is a great one. I'd recommend you check out this blog post if you are new to Nextflow and you want to learn about this. So this is 23andMe, and they've got a great post from their engineering team actually about why they use Nextflow, particularly for their massive scale up. So this is if you look at this blue line here in the middle of the diagram, you'll see the number of CPUs in a computer environment. So this is up to 10,000 CPUs running in a single, in this case, AWS batch environment here. So Nextflow is really running that, orchestrating that. If you look at the top line there, you'll see the number of tasks, and you'll see that they've had, within a 24-hour period, something in the order of 865,000 tasks. That means those tasks, which are, each task is a containerized job. So orchestrating all of that at scale is, it's possible here, and it's kind of something which is we really look towards as we think about enabling scientists to use that scale and kind of use that power. I'd also point out that not everything works first time, and I think there's a great example where we showed before on the example of the errors and what happens when those errors happen. Well, ideally you want to debug, but sometimes it's just, if you run a million jobs, those things will happen. The ability to automatically re-execute a task, automatically re-execute a pipeline, basically using that infrastructure just removes a lot of hassle without having to do a whole bunch of recompute, and again, kind of saving a lot on the cloud costs of that. If we look at other areas, one big area that we've seen a lot of adoption is in the personalized medicine space. So CAR T therapies, anywhere you have a lot of sequencing. This is an example here of the BioNTech treatment which has been licensed to Genentech here. In this case, it's for phase 2 melanoma, essentially taking a sample of normal tissue and also tumor tissue, identifying the neoantigens on the surface of those cells, really kind of the differences, using some machine learning, and then really creating a personalized mRNA against this. Obviously, these therapies are very expensive at the moment, but it's kind of great to see that there's the use of this technology really converging high throughput sequencing with the ability to manufacture very targeted, personalized vaccines and cell therapies. It's obviously a huge area of research and something that we kind of see adopted by many of our customers as well. And just finally touching on the kind of the use of genomics and as part of routine screening. This is something which is becoming more and more common, and we start to see this in customers like Invitae, the largest sequences around the world, and using that information to kind of drive outcomes and help patients here. And again, so we've got this whole spectrum from iterative research of where you're really changing pipelines, changing code, changing experiments every time, all the way through to this area where we're doing high throughput kind of routine sequencing, routine analysis here. And it's not just sequencing, it goes into imaging as well. The use of AI and ML, particularly the use of GPUs and FPGAs, things like Dragon, become really kind of significant, particularly at scale. And if you're kind of thinking about those costs and how to optimize those things and really integrate with the rest of your system. So with that, I will pass it over to the question time. If you've got questions, folks, we'll answer those. And thank you so much for joining. I'm looking forward to answering a few questions.
Thank you, Drs. Ahdesmaki and Floden. As a reminder to webinar participants, if you have a question, please type it into the Q&A box in the control panel. We'd like to ask attendees to take a moment after the webinar has ended to take our exit survey and give us your feedback. We'll now start the Q&A portion of the webinar. The first question is for Dr. Ahdesmaki. Relating to Seqera's fourth, what are the kinds of productivity increases that you've seen at AstraZeneca? the kinds of productivity increases that you've seen at AstraZeneca resulting from the use of Nextflow Tower, and automation?
Yeah, no, that's a great question. It's about what kind of time saving or productivity improvements we've seen. And I would split that into two. So there's what you just couldn't do, and then what would take less time. So what I alluded to so let's say 3 years ago, or 4 years ago, when we started on this path to make this more automated, the way to run this CRISPR pipeline would have been more. There would be the initial Nextflow implementation, but the output would be a bit more like, let's say, CSV files, and you would spend days trying to wrangle the information from the lab into these CSV files and in the right formats. So it would have taken several days probably to launch the pipeline and then make it run, and then it wouldn't go to the database because it wasn't there and there was no automation to get it. So I would say that just in this simple example of this running these pipelines, you save days of the manual effort, and you can use all of that time because it's all automated to do something else, or just basically do more downstream interpretation analysis of the results. And I'm sure there's other examples in other areas, but I would say in this very specific CRISPR example, it saves just the analyst saves days of time.
Great, thank you. The next question is for Dr. Floden and Seqera. Could you elaborate on how Seqera's architecture accommodates scalability to meet the computational demands of both small-scale and large-scale products?
I think it's one interesting thing to point out there is like when we were running Nextflow, or even the installation of Tower and how it's connecting in with your resources, it's running in your environment. So that can be the environment that you set up within AWS or Azure or Google Cloud, but it can also be environments that you have set up with on-premise schedulers. What it's enabling you to do is just have visibility on those resources. And particularly, we've seen in cloud environments, the ability to scale. So if you are, for example, running a pipeline where the data comes off a sequencer on Monday, and it takes a couple of days to get that data processed, to kind of automate, go through these pipelines, as soon as the pipeline analysis is finished, let's say on Tuesday at lunchtime, that compute will shut down, and you will essentially not pay for anything after that. The compute is kind of spun up for that moment and shut down. And that principle kind of applies all the way through. When we look at particularly bioinformatics workloads, they're very spiky. They have periods where they need a lot of CPU, where they need a lot of memory, where they need a lot of GPU. And the ability to have this kind of scalability kind of going up and down is very important on a cost saving perspective. Just on this kind of scale thing itself, this is where we've been building some technology called Fusion, which is really interesting in that it allows you to mount object storage inside of those virtual machines. Slightly kind of technical topic, but when you think about the type of information that we're dealing with in this space, it's often large compressed text files of just large amounts of genomic information that needs to be moved around for each task which runs. And I showed you before that sometimes with pipelines with tens of thousands of tasks, that movement and that kind of need to be able to spin that up, move that around is quite taxing. It's also quite difficult to achieve. It's quite different to many other kind of workloads that exist and are kind of running on cloud. So what we've been building is the ability to kind of do that in a manner which enables you to sort of happen without having to wait for that job to finish, without to wait for the job to run, and without needing to spin up specific kind of compute storage to run that. In that case, we're looking at some of our customers who are running very large workloads who are saving 50% straight off the bat and using that technology. And I think particularly as you go to scale, that's the kind of thing that we're investing a lot in to be able to and really just enable them to do stuff, really enable them to do harder science.
Great, thank you. Another question for you here is, given the sensitive nature of genomics data, how does Seqera ensure robust data security and compliance with international regulations?
Yeah, that's another way. It very much relates to what I was mentioning before. So when you are running really in your environment. And I think we heard about it in the first talk where we're talking about the analysis itself is taking place within AZ's firewall. So this gives you the full security and the full, you know, you're not even relying on anyone else to do anything for you. You just have that by default by running in that environment. It doesn't need to move the data. You're moving the analysis to where the data is already, and that's a really important point. It's something which we see particularly large customers kind of want, that they are becoming more sophisticated with their cloud setups, and that they really want to have control, understand the boundary that exists in that data, and to be able to really kind of manage that without having to rely on third parties to do so.
Great, appreciate it. Another Seqera question: how is Seqera fostering the principles of open science to facilitate collaboration and information sharing within the scientific community?
That's great. I think it kind of goes back to our origins, and as I said, we started this as people who wanted to be able to develop pipelines and share them. Some of the really important projects in the ecosystem, like NF-Core, came out of really organic conferences that we were holding, and really from that was the idea that people wanted to be able to not duplicate their work. They didn't want to necessarily need to write pipelines 3 times or once for every different infrastructure that they would be using. Also, we could start to bring domain experts together. If you look at NF-Core, it's got something like 70, 75 pipelines which are released now. Each one of those pipelines has been really developed by domain experts who understand the technology which the data has been generated from, the tooling which it comes from, the analysis which is taking place there. And that knowledge is just something that, as an Nextflow user, as someone who uses, it's part of the kind of open science ecosystem, you just simply instantly lock into that, and you can really leverage that for your own work. That means you focus on your data and your science necessarily than having to read that as well. I think more broadly, this is something which is taking place across many areas. If we look at some places in machine learning models, for example, we're starting to see this proliferation inspired by the use of Git and sharing, and this ability to really have science happening in the open, where we really all benefit from. And that's something that we believe in, that we kind of strongly lean into, and are really excited about where it's going to take the future.
Great. And one more question here, also for Seqera. Could you share some success stories or use cases where Seqera's modern biotech stack played a pivotal role in accelerating scientific research and discoveries?
Yeah, there's been a couple of these where it's been really significant, and I think one that's been great is an Arcus, and they are a biotech which has been scaling very rapidly, had some really interesting pharma collaborations as well as they've scaled out. When they look at their stacks and what they were doing before, within 12 months of working with us, they're really saving sort of 40 to 50% on those cloud costs, as well as being able to kind of pull up, be able to automate a lot of the things. And much of the stuff that we saw in the diagram before, where we were connecting to QC services, we were connecting to your databases, where you're being able to build in the business intelligence on that, the automations, that's really the stuff where we're seeing customers adopt, and that we can pull more and more into using integrations, using open source tools, using other platforms as well to integrate with, without having to spend so much time kind of building everything yourself and doing a lot of the plumbing. It's kind of a lot of the principles of how Nextflow started, and it's kind of things that we're applying now into the Seqera platform.
Great, appreciate it. For Dr. Ahdesmaki, why did you choose Nextflow as your workflow manager instead of some of the other choices out there?
That's a reasonable question. So why Nextflow as opposed to something like SnakeMake or building our own? There's a few factors to that. So let me go back several years. So I've been involved before Nextflow was around in building pipelines, and I was a heavy contributor to something called BC Bio NextGen, and it was an effort from the people at Harvard to build NGS, reproducible NGS pipelines, and there was really nothing much out there. But they kind of, what you learn along the process, if you build something yourself, eventually it becomes quite difficult to maintain something. And with Nextflow, the great thing is that it's like what Evan said, is like a lightweight wrap around what you already have. And then there's the NF-Core community where you can get putting this in. So I think there's the ease of use for Nextflow, but also the familiarity of certain people in building Nextflow pipelines that for this specific functional genomics application, it may help us make the decision. And we haven't really looked back because it works so well. And with the updates to Nextflow, the modularization, what they call the language specification too, and going forward helped us. And then also the Nextflow Tower itself, because it nicely sits on top of Nextflow. So it's kind of multiple factors that all came into one, and that helped us with that decision. Hope that answers the question. I'm happy to kind of elaborate if there's any other parts to that.
Great, thanks. It appears that is all the questions that we have for now. We'd like to thank Miika Ahdesmaki and Evan Floden and our sponsor, Seqera Labs. If we didn't have time to get to your question, or if a question comes up later, we'll try to follow up with our experts. And as a reminder, please look out for the survey after you log out to provide your feedback. If you missed any part of this webinar or would like to listen to it again, an archived version will be mailed to all attendees. Thank you for joining us for this Genome webinar.